I am standing here with the neuron that we have been examining most of the semester, Ned the neuron, our familiar guy. Um, and I want to use Ned to talk about graded potentials. Uh, so um, First of all, before we dive in, I want to do a very quick review of resting potential, which we learned about in prior weeks of the course. And, you guys displayed your understanding of that topic on the last quiz. So this will also go over some of the content from our prior quiz. So during resting potential, when the neuron has nothing to say, inside the cell, there are some large protein particles that are trapped inside the cell. I compared them to the furniture in an apartment that's too big to get out the door. These particles can't leave the cell through any of the gates and channels on the membrane. My question is, are these particles positively or negatively charged? Who remembers?
Negative.
Just shout it out. Negative, yeah. So we have these big negatively charged protein particles, and their presence inside the cell is what really accounts for most of resting potential. Because in resting potential, remember, the cell is more negative on the inside. So what other ion is concentrated inside the cell when the cell is at rest?
Potassium.
Absolutely. Okay, great. And then what ions do we see concentrated outside the cell when the cell is at rest?
Sodium.
Yes, sodium. And we said that sodium and potassium were sort of like the co-stars of the movie that is the action potential. But we do have a supporting cast member that influences graded potentials. Does anyone remember that supporting cast member?
Chloride.
Chloride. And where is it concentrated when the cell is at rest? Inside or outside?
Is it outside?
Yeah, okay, great. So here is our picture of our cell during resting potential, which, remember, is the state of readiness that allows the cell to be ready to send a message as soon as the neuron decides to fire. So we're contrasting action potentials and graded potentials today. So hi, I blanked on my question. Okay, so who can tell me which of the two is the big all or nothing change in charge that zips down the axon when the neuron has something to say? Is that the action potential or graded potentials?
Action potential.
Yeah, that's right. So, and the name says it all. Action. The action potential is what happens when the neuron has something to say and a huge change in charge whips down the axon and it carries the neuron's message to other cells. Graded potentials are more subtle. They're gentle changes in charge that help the neuron make this all-important decision of whether or not to fire. So we can think about it this way. I'm going to go ahead and take the protein particles out because we kind of know where they are all the time. And so we'll kind of put these here. Okay. Oh, I'm missing a pin. Just a sec. Okay, sorry, I'm really living that chaotic professor or absent-minded professor stereotype today. Just a sec, sorry. Okay, thanks for your patience. Okay, so within the cell, graded potentials happen in a particular area and action potentials happen in a different area. So we're going to divide our neuron into 3 zones for the 3 parts of neuronal communication. And So in this sort of very last zone, this is our neuron Ned. And way down here, we have Ned's terminal buttons, where Ned secretes neurotransmitter when it's time for him to send a message. So we're going to talk about this part of the process, the chemical communication between neurons, more as we get into next week. But today we're talking about electrical communication within the neuron, graded potentials and action potentials. So where in the neuron do graded potentials occur? In the axon or in the cell body and dendrites?
The cell body?
Yeah, that's right. So graded potentials happen out here in the cell body and the dendrites. And action potential happens in the axon. So the two types of electrical activity take place in physiological distinct regions of the cell. And the whole idea of graded potentials is to help the neuron take in positive and or excitatory and inhibitory messages and decide whether or not it's going to fire. And this language of excitation and inhibition is crucial to understanding everything we've talked about in this course so far. I'm reaching over here to grab a student question. So this question came to us from Chelsea, and she asked, how do specific and specialized systems of the brain integrate with one another? And this is a really good question, because for the whole first part of the course, we learned about specific brain regions that do particular functions, such as the prefrontal cortex that controls logic and reasoning, and the amygdala that fires up emotions like anger or fear when we detect a threat. And we've kind of alluded to the idea that these brain regions can communicate, which would be integrating with one another. But we've never really gotten into the biology of how that communication takes place. And that's what this week is all about. When we say that the prefrontal cortex influences the amygdala, what we actually technically mean is that neurons that have their cell bodies and dendrites in the prefrontal cortex have long axons that project down to the amygdala and dump out neurotransmitters in the amygdala that affect it electrically. They either raise the charge inside cells in the amygdala, exciting them, making them more likely to fire, making you more hyped up and ready to get angry or fearful, or they can secrete neurotransmitters on the amygdala that lower the electrical charge in neurons inside the amygdala, inhibiting them, quieting down electrical activity there, making the amygdala less likely to fire. So this inhibition and excitation is what allows these brain regions to communicate in the ways that we've been discussing all course. So it's very important. So let's take a little bit of a more close biological look at how inhibition and excitation actually happen in an individual neuron. So when we're talking about a neuron inhibiting another neuron or exciting another neuron, that's communication between two brain cells. So I've got to introduce another character. We can't tell the whole story just with Ned here. So I've got another neuron that I'm bringing to the table. This is an excitatory neuron, meaning that when it dumps neurotransmitter onto the dendrites of Ned the neuron here, our superstar, it makes Ned more likely to fire. So this is, I'm going to put this neuron kind of up here so that its dendrites, or so here, its dendrites and cell body are up here. And when this excitatory neuron fires an action potential, it dumps neurotransmitter onto the dendrites of Ned the neuron. So, it can get a bit confusing as we've got multiple cells. So neuroscientists have a convention about how to refer to two cells in a chain of communication like these two. They call the neuron that is sending the message in question the presynaptic neuron. And that's because it exists before the synapse, which is simply the gap between the terminal buttons of the neuron that's talking and the dendrites of the neuron that's listening. So in this example, this screen excitatory neuron would be the presynaptic neuron, and Ned himself would be the postsynaptic neuron because he is listening to the neuron up here. Now, if we change our focus and we start to discuss what happens when Ned sends a message to a third neuron that exists out this way, when Ned is dumping neurotransmitter onto the dendrites of another neuron, then the frame of reference changes. Ned becomes the presynaptic neuron, and the neuron that Ned is communicating to becomes the postsynaptic neuron. So that can be a little tricky to grasp, but it's actually a really handy way of referring to connected neurons, kind of like left and right are in reference to the body. Presynaptic and postsynaptic are in reference to the direction of the message. So the postsynaptic neuron is listening, it's receiving neurotransmitter, and its electrical charge is getting changed, affecting its decision as to whether or not to fire. And so that's the postsynaptic neuron, and then the presynaptic neuron is sending inhibitory or excitatory messages. Questions so far? I'm throwing a lot at you. Is it making sense so far? Okay, so if this neuron is excitatory, we know that it makes Ned, our star neuron, our postsynaptic neuron, more likely to fire. So what does that actually mean? We know that the neuron during resting potential is a little more negative inside, but if the charge reaches a certain point called threshold, then an action potential will fire. Does anybody remember the specific voltage that is threshold, that magic number where the action potential gets started?
Is
Any
it
guesses?
55?
Yeah, negative 55 millivolts. So remember, at resting potential, the cell's hanging out at negative 70 millivolts. So the charge right here at this borderline between the cell body and the axon, this special region right here, has to raise all the way up to negative 55 millivolts in order for the action potential to fire. So in this special area where the decision of whether or not to fire gets made has a particular name. Does anybody remember what this special area is called?
Axon hillock.
Yeah, axon hillock. And in the video, if you got the chance to watch it, I put a picture of Count Von Count there because this is the location where the neuron is sort of counting the votes. So chemically, how do those votes come in? I've already said in the class, when an excitatory neurotransmitter, or when an excitatory neuron releases neurotransmitter and it's picked up by a postsynaptic neuron like Ned that's listening to this excitatory message, it changes the charge inside the cell. Let's get potassium out of the way. Changes the charge inside the cell in a way that makes the cell more likely to fire. So would this excitatory message raise or lower the charge inside the postsynaptic neuron if it's making it more likely to fire? So resting potential is what? When the neuron's at rest, what is the charge inside the cell compared to the outside?
Negative 70.
Great. And we just heard that threshold potential is negative 55. So when the charge changes from negative 70 to negative 55, then the neuron fires. So if an excitatory connection makes the neuron more likely to fire. So does it do that by raising or lowering the charge? This is a little math question.
Raising it.
Lowering.
Okay, First of all, that was great. I got a whole choir of voices and I heard raising and lowering. So let's talk it through. So we at rest, it's more negative inside the cell. The charge has to increase from negative 70 millivolts up to negative 55 millivolts for an action potential to fire. So an excitatory connection has to raise the charge inside the cell. So if this cell wants to influence Ned's decision, this cell wants to excite Ned to make him more likely to fire. It has to raise the electrical charge inside Ned, hoping to make it to the axon hillock and boost the charge there enough to reach threshold and cause Ned to fire. So the way in the past, at this point in the lecture, students have asked me a really smart question. They said, Dr. Cook, what makes a neuron excitatory or inhibitory when it's communicating with another neuron? Is it the neurotransmitter it secretes? What is it? And the answer is, it depends on the type of ion channels that the sending neuron, the presynaptic neuron, opens on the dendrites. So at the synapse, the presynaptic neuron, the one that's sending the message, dumps neurotransmitter onto the dendrites of the postsynaptic neuron, the one that's picking up the message. And that neurotransmitter will learn, opens up channels in the membrane of the cell that allow ions to pass in and out. So when we studied resting potential, we saw that it's really important that every ion has its own door to the cell. I showed you a little house with 4 different goofy doors lined up. So we know that there's certain gates that sodium can go through, there's certain gates that potassium can go through, and they are, opened and closed under different conditions. So a very important biological distinction between the 2 areas of the cell is that in the cell body and dendrites, where graded potentials take place, the channels on the dendrites only open when neurotransmitter affects them. They're neurotransmitter sensitive. The channels, the ion channels that allow ions to flow in and out of the cell down in the axon, open for a different reason. And I'll talk about that later. So these neurotransmitter sensitive channels open up, allowing the electrical charge inside the receiving cell, the postsynaptic neuron, to change in a way that makes it more likely to fire. So my question is, we've got sodium and chloride on the outside, and we've got potassium on the inside. If this cell wants to make Ned the neuron more likely to fire, to raise the electrical charge inside Ned, hoping to reach that magical threshold point, which ion should it allow to pass more freely? Does it need to open those sodium gates? Does it need to pry open the door for chloride and get it flowing? Or does it need to open the potassium gates? Which gate being opened or opened further would raise the charge inside Ned?
It would have to open up for sodium.
Okay, great. And so why do you say that? What would happen if the sodium gates on the dendrites or cell body opened up?
So sodium has a positive charge, and entering would help positively charge the inside of the cell.
Yeah, that's great. So remember we said that the sodium, the electric force and the diffusion force both push sodium to want to enter the cell, but the gates on the cell body and dendrites and axon are all locked tight. They don't let that sodium in. It is packed up outside the cell like Black Friday shoppers hanging out outside of Walmart. And in fact, it's some sodium sneaks in, and we've got the sodium potassium pump pushing it out. So it's like a crush crowd of sodium waiting to get into the cell. So an excitatory neuron, all it has to do is dump out a neurotransmitter that opens sodium gates, and a huge flood of sodium, like a stampede of wild horses, enters the cell, raises the charge in the dendrite, and makes the neuron that's receiving the message, the postsynaptic neuron, more likely to fire because its charge has increased now. It's a small fluctuation, so maybe it goes up from -70 to -68 or -65. But if enough of these excitatory connections were coming in from different neurons at the same time, and they all fired at once or close together, a lot of excitation, then a wave of positive charge would reach the threshold point, and it would actually trigger that action potential. So it's the movement of sodium that allows in an excitatory connection to increase the charge inside the postsynaptic neuron and trigger an action potential. But inhibitory connections aren't the only ones that are important in the brain. Or excitatory connections aren't the only ones important in the brain. We've talked a lot about brain regions inhibiting each other, calming each other down. So for instance, we talked about how in a moment where you're beginning to panic, sometimes as the amygdala gets hyped up and gets ready to activate the hypothalamus and kick off fight, flight, freeze, sometimes the powerful prefrontal cortex can kind of take command, steer the ship, and it sends calming messages to the amygdala. And that happens when you think thoughts that sort of take your emotional temperature down a peg and allow you to function in a more reasonable way. So if you're about to fly off the handle and you use your mind to calm your emotions, what is happening inside your brain is that neurons in the prefrontal cortex are dampening electrical activity in the amygdala. So they're making cells in the amygdala less likely to fire. These inhibitory connections work a little bit differently. So now we're not talking about an excitatory connection. We are imagining a neuron that has inhibitory connections that's dumping neurotransmitter onto NED, that's opening ion channels that make NED less likely to fire. So our excitatory connection raised the voltage inside NED, getting him closer to threshold, making him more likely to fire. So inhibitory connections coming into NED are going to lower the charge inside NED, lower the voltage, bringing it even further from threshold, making it dip below resting potential, and making it all the less likely that he's going to fire. So how would an inhibitory neuron achieve this? What channels or gates could it open to lower the electrical charge inside NED, making him less likely to go off, less likely to fire? Would it be sodium, potassium, chloride, more than one? What's your guess? Let's think through it. We know that potassium's usually free to come and go, and it mostly hangs out inside the cell. What would happen if something caused a bunch of potassium to rush out? Would the charge inside the cell stay the same, get higher, or get lower if a bunch of potassium leaves at once?
Get lower?
So potassium is a positively charged particle, and when a bunch of potassium floods out of the cell, that is correct. That it will leave a negative charge behind. A bunch of positive ions flowing out lowers the charge inside the neuron. So one way that an inhibitory connection can work is by opening those potassium channels a little wider and causing potassium to shoot out of the cell. That brings the charge down even below resting potential and makes the main neuron we're talking about, the one that's listening to the message, the postsynaptic neuron, less likely to fire. This can also be achieved by opening up the chloride channels and bringing chloride in, because it's a negative particle. So when it comes in, it lowers the charge inside the cell. So the neuron is listening to hundreds and sometimes thousands of other neurons through its dendrites. So this is a simplified drawing, but you should imagine a real neuron, unlike Ned here, who has, I've drawn you 4 dendrites. A real neuron has a whole forest of dendrites with hundreds of incoming connections that all deliver tiny excitatory or inhibitory messages to the cell, tiny changes in voltage. And in the axon hillock, where it all comes together, it's sort of like Washington, D.C., where the votes are counted in the United States. And enough excitatory messages will raise the charge and make the cell fire. But if a ton of inhibitory messages are coming in at the same time, they may block that excitatory charge, sort of like the goalie standing in front of a soccer goal can block the ball from coming in. A well-placed inhibitory connection can lower the charge at the axon hillock in a way that kind of cancels out a wave of excitation. And this would happen when imagine a cell in the amygdala that's getting a lot of excitatory input, probably coming from that shortcut we've talked about that brings information from the thalamus that's emotionally relevant straight to the amygdala, so you can fire off a panicky response before your brain's even processed the scene. So let's say that the thalamus is sending a ton of excitatory input to the amygdala and charging it up. If at the same time you're really focused on staying calm, the prefrontal cortex might be sending an inhibitory input to the amygdala, and that is you focusing on trying to control your emotions. And it's sort of a battle inside the neuron between all the excitatory influences and all the inhibitory influences as to which wins in a given moment. Does the neuron fire or not fire? And we're going to take a 10-minute break now. And when we come back, I'll review this content in a slightly different way in case you're still feeling fuzzy. And then we'll talk about what it looks like when the action potential is actually fired. What is it that allows the electrical message to travel down the neuron to the synapses and send that yes or no message? So let me see, it is 2:03. So we will come back at 2:13 to talk about action potentials.